we'll be talking about how endoplasmic reticulum and Golgi operators are interrelated. Now, first observe the diagram, then I'll explain the points. See, this is the nucleus, small part of nucleus they have shown in this diagram. I'm just circling on that. So this you can take as the nuclear membrane. Now from the nuclear membrane, the endoplasmic reticulum will stand. So this is the endoplasmic reticulum, the tubular structure. I'm just shading on that. So this is the endoplasmic reticulum. And based on the presence or absence, we can differentiate. Here we can find clearly dark ribosome bodies present on the endoplasmic reticulum. So it's definitely RER, rough endoplasmic reticulum. Now rough endoplasmic reticulum is involved in synthesis of proteins. So here the proteins are produced from the endoplasmic reticulum, this part. Now produced proteins will be pinched off. So they'll be removed in the form of a transport vehicle, which we call as vesicle. This transport vehicle we call as vesicle. Okay. Now this vesicle, which is containing the synthesized proteins, will be accepted by the Golgi complex. So as I told, nuclear membrane in continuation with endoplasmic reticulum, which will be in continuation with Golgi complex. See, this is the pattern. Now when you take the Golgi complex from the endoplasmic reticulum, the proteins will be transported through vesicles. Now it will be taken from the cis side of the Golgi complex and packaging happens inside the Golgi complex. And once the proper target is added inside the Golgi complex, they will be moved towards the transphase. So this is how the vesicle with proteins packed in it will be removed towards the cell membrane. So this is the cell membrane. So that is the order. When you take the order, you'll have the nuclear membrane, which will be in continuation with endoplasmic reticulum, which will be in continuation with Golgi complex. That will end up with cell membrane. So this is kind of flowchart you have to remember. So why it is happening in this pattern only, that is explained over here. See, the ribosomes, which are there on the rough endoplasmic reticulum, they'll assemble proteins. Assemble proteins here is nothing but a polypeptide will be taken up and proteins will be produced. Poly, many peptides, amino acids. Many amino acids will be clumped together to form a protein. This is the basic idea behind molecular biology you have to know right now at this stage. Okay. So the ribosomes on the rough endoplasmic reticulum, what they will do? They will synthesize proteins. How proteins are synthesized? That point I am explaining over here. How proteins are synthesized? Proteins are synthesized by assembling polypeptides. Polypeptides is nothing but many amino acids will be assembled to produce proteins. So once the proteins are produced, the proteins produced will be moving through rough endoplasmic reticulum, where they are further modified, and through transport vesicles they'll be pinched off. Pinched off is nothing but they'll get removed from the endoplasmic reticulum, rough endoplasmic reticulum, and they'll fuse with the membrane of Golgi complex. So that's what I have shown here. In the earlier example also I showed the same slide. So in the Golgi complex, the transport vesicles will come, and within the Golgi complex, remember the word, within the Golgi complex, the proteins are further processed and stored. What does mean by processed here? Processing is nothing but adding sugar molecules or adding any ions. For one example, I take sugar molecule. For some other molecules, you'll have adding of ions. Okay. So this is all called processed. In other words, we said packaging, isn't it? The same happens in Golgi complex. Now once the proteins are finished off packing properly to go to their destination, they'll be again pinched off from the Golgi complex and they travel towards the cell membrane. And they'll release the proteins towards the outside. So this is how the proteins will be produced, packed, processed and released to outside. So that's the flow chart. From the endoplasmic reticulum towards the Golgi complex, towards the cell membrane, they will be removed towards the target part. This whole process we call as protein processing and secretion. Generally, they'll give a word, a crude word like this. So what does mean by protein processing and secretion? Simple. Protein processing and secretion is totally related to production of proteins by endoplasmic reticulum and proper packing of the proteins in Golgi complex, then transport of these processed proteins towards the target part out of the cell membrane. This whole thing we call as protein processing and secretion. It's okay. Anyway, so here when you take the Golgi complex, it is in continuation with endoplasmic reticulum. First point. So here you will have endoplasmic reticulum. And Golgi complex other side will be in continuation with cell membrane or plasma membrane of the cell. So this is the order. Why the same order has to be followed? That will be explained in this slide. So here when you take the nucleus, from the nuclear membrane, endoplasmic reticulum is there, rough endoplasmic reticulum specifically. Then it is in continuation with Golgi complex. Then Golgi complex is in continuation with the cell membrane. Now, when we take, excuse me, when we take the protein processing and secretion concept, this is the basic function related to endoplasmic reticulum and Golgi complex also. So how both the cell organelles are involved in this function aspect. Now first what happens? In the rough endoplasmic reticulum, you will be having ribosomes. Now what ribosomes will do? Ribosomes will produce proteins. How proteins are synthesized? Proteins are synthesized by ribosomes by assembling polypeptides. Polypeptides here stands for poly, many. Peptides are nothing but the amino acids joined together. So proteins are synthesized by ribosomes by assembling many amino acids so that protein will be produced in rough endoplasmic reticulum. This is the first point. Once proteins are produced in the rough endoplasmic reticulum, from there they will move through transport vesicles. Vesicles are the part of endoplasmic reticulum. They'll be pinched off here. Okay, they'll be pinched off. Pinched off is nothing but they'll just get removed from one part and it go to another part. So they'll be pinched off from the rough endoplasmic reticulum, the vesicles, which will transport proteins. The proteins are produced, which will be transported through these vesicles from rough endoplasmic reticulum towards the Golgi complex. So this is how the order. From endoplasmic reticulum towards the Golgi complex. So in the Golgi complex, the proteins are processed and stored. So processing here is nothing but the packaging. Packaging is nothing but adding sugars to the proteins so that they'll go to particular destination. In some examples of protein molecules, not only sugars, some ions also will be added. So this adding extra sugar or adding extra ion molecule to proteins is nothing but processing. In other words, we can say as packaging, because in some textbooks they are using this word also. Packing of proteins, where it will happen in Golgi complex. Processing of proteins, where it will happen in Golgi complex. So first point, proteins are synthesized. Second point, from the endoplasmic reticulum, they will be transported. How they will be transported? Through transport to vesicles by pinching off from endoplasmic reticulum. And in fourth point, the transport vesicles will deliver the proteins towards the Golgi complex. And what is happening in fifth point? The exact processing of proteins. Okay. Then in the next step, these finished proteins. What are the finished proteins? The proteins which are processed, which are packed with sugars. They will be pinched off. They will be pinched off from the Golgi complex. See here. So this is one stage from endoplasmic reticulum towards the Golgi complex. This is the second stage from Golgi complex towards the cell membrane. Okay. So 2 times the transport vesicles getting pinched off from endoplasmic reticulum and from Golgi complex second time. After finished proteins, they will be pinched off from the Golgi complex. Now they will fuse with the cell membrane. Okay, that's the 7th point. So what is happening? The vesicles, they're traveling from Golgi complex towards the cell membrane, isn't it? From Golgi complex towards the cell membrane. And they'll fuse with the cell membrane like this. See, I'm showing on the diagram. Maybe I'll use some different color. Here. Here, this is the transport vesicle with protein. I'm just shading on the diagram, just observe it. So this is the plasma membrane, isn't it? So from the plasma membrane, they will fuse with the plasma membrane, the evagination of plasma membrane, and they will be released outside. So the curved structures are nothing but the packed proteins. So that is nothing but secretion of proteins. So this is the whole process of protein processing and protein secretion, which will happen in endoplasmic reticulum and Golgi complex. Specific term related to Golgi complex is processing of protein. This is specifically related to Golgi complex.